The TROPICSAFE project, funded by the European Union's Horizon 2020 Research and Innovation Program, addresses 3 economically important insect-borne prokaryote-associated diseases of palm, citrus, and grapevine in tropical and subtropical areas. Over the last decades, these are the diseases most seriously affecting the worldwide trade and import of products and materials from these crops because of trade globalization and climate change. These diseases are due to the presence of diverse Candidatus Phytoplasma species associated with lethal yellowing in palms and yellows in grapevine, and Candidatus liberibacter associated with Huanglongbing in citrus. For their effective, efficient, and economically and environmentally sustainable management, the project filled important knowledge gaps covering Africa, Ghana, South Africa, and Mozambique, America, Mexico, and Chile, the Caribbean, Guadeloupe, Jamaica, and Cuba, Europe, Spain, and Italy. Take a tour around the globe with us to learn about the main findings of the TROPICSAFE project. It is a trip organized by Crop. Hop on! Outbreaks of lethal yellowing are threatening the coconut industry economy and the livelihood of millions of rural inhabitants, especially in Mexico, the Caribbean, and Africa. The disease also has serious environmental repercussions related to soil degradation and negative effects on species biodiversity. Several different phytoplasmas are involved in coconut lethal yellowing worldwide. The project surveys in Africa resulted in the identification of strains of this phytoplasma in coconut palms in Ghana and in Mozambique. In Ghana, evaluation of 5 dwarf coconut varieties allowed to identify some with superior growth in heavily infected areas. This phytoplasma group was detected in coconuts in Cuba, Jamaica, and Mexico. The plant hopper, Haplaxius crudus, was confirmed as the main insect vector in Mexico, while in Jamaica and Cuba, other possible insect vectors were identified. In Jamaica, 2 weeds resulted positive for this phytoplasma group. In Mexico and in Ghana, different palmicotypes are under field evaluation for resistance, also using specifically developed molecular markers. For the identification of these phytoplasmas, a specific LAMP diagnostic and a multiplex PCR were developed, validated, and applied. Grapevine yellow causes serious economic losses worldwide. In the countries studied, this disease is associated with different phytoplasma. This phytoplasma was detected in South Africa in the following plants and insects: in these vineyards, it was also found the abundant presence of the leafhopper, vector of this phytoplasma. The management plan developed to treat grapevine yellow disease in South Africa includes recommendations for leafhopper monitoring, weed control, plant sanitation, and chemical control. Plant-derived antimicrobial peptides were prepared for preliminary screening against phytoplasma containing colonies. LAMP and qPCR assays for specific detection of the phytoplasma strain from South Africa were tested. In grapevines in Chile, different alternative host plants for this phytoplasma were identified. And the following insect vectors or potential vectors. In Italy, 16 plants and 12 insect species collected in vineyards were positive for the presence of diverse phytoplasmas. These were the main phytoplasma disease targets in Italy for management and detection. An experimental vineyard obtained with the F1 crossing population between genotypes with differential susceptibility to flavescence dorée was infected using insect vectors. 300 biotypes were genotyped and showed about the 20% of the progeny as self-crossed. Sequencing to identify genes putatively associated with the susceptibility or resistance of varieties with opposite phenotypes was performed. Analytical performances of the developed serological protocol for phytoplasma detection were evaluated comparing ELISA versus quantitative PCR. The antiserum produced using this phytoplasma-containing colonies was successfully employed in immunofluorescence assays. Huanglongbing is the most destructive disease of commercial citrus species worldwide. Its impact is very high in America and Africa, and one of his insect vectors is present in the Iberian Peninsula, the main citrus-producing area of Europe. The Candidatus liberibacter species are associated with this disease. The surveys in citrus confirmed the presence of the agents in South Africa, Guadeloupe, Jamaica, Cuba, and Mexico. In South Africa, a specific quantitative PCR was established for the detection of Candidatus liberibacter africanus. The presence of Chirioza erythrae was confirmed in South Africa and in Spain. The project could also confirm the presence of the Asian citrus psyllid in Cuba, Mexico, and Guadeloupe. The surveys did not result in the detection of alternative insect or plant host species. Since native parasitoids of this psyllid were not identified in Spain, a parasitoid from South Africa was introduced and showed good dispersion of parasitum efficacy in the Canary Islands first and in the Iberian Peninsula later. The seasonal trend of Chirioza erythrae determined in the Canary Islands indicated between 4 and 5 generations per year, but its population remained very low after the summer. The project developed predictive models that estimated the invasion risk of this psyllid in the citrus growing areas of Europe, Near East, and North Africa. A survey confirmed the presence of Diaphorina citri in the main citrus-producing areas of Cuba, with a seasonal trend connected to management strategies and irrigation or rains. Removal of symptomatic trees at a regional scale proved to be the best management strategy, and kaolin treatments kept the infestation level very low for 2 years. In Guadeloupe, despite the relatively low abundance of the vector in orchards under an integrated pest management program, the disease levels and mortality of the citrus trees were very high. Different combinations of rootstocks and varieties were selected, and after 4 years, all the trees became infected, although they were asymptomatic and had fruits. The quantitative PCR analysis showed a low concentration of the bacterium and verified the presence of a relationship between Huanglongbing susceptibility and anatomical, physiological, and metabolomic traits, with a special focus on the potential impact of polyploidy on disease tolerance. This video covered the main outcomes of the TROPICSAFE project, product of a collaborative research effort of 22 partners from 12 countries working together from May 2017 until April 2022. Much more information is available on the project website. Don't be shy and visit us.